Everyone. I'm Tony Latai, president of the Society for Functional Precision Medicine, and welcome to 2024. Just a couple of minor things, maybe not minor things, but a couple things before we get started. First, this is the beginning of the year. You have gotten a mailing and will probably get additional ones. This is a great time for you to renew your SFPM membership. You do need to renew it annually. And this year, in addition to all the benefits you've had before, there will be some perhaps more tangible benefits because it will include discounts to the big meeting coming up in Copenhagen in September. So it is well worth your while to renew. I'll also say it is very important for the strength of this society to keep a solid membership base. So please do your part and re-up as a member. And this also goes for people for whom membership dues are zero, trainees. Please make sure your membership is current. I would also like to take this moment to thank our sponsors, particularly Cayenne Technologies, but also Travera, Presage, Exscientia, Kiatech, Zillis, and 2Curex. Thank you very much for your financial support. We run a very lean organization, but there are still some costs that are inevitable with regard to running a society, and we really do appreciate your absolutely necessary support. So today we will have two speakers, both from St. Jude's, Kristine Cruz and June Yang. Kristine Cruz has for some time been Director of Research Operations at St. Jude's Children's Hospital with a particular interest in pharmacogenomics, and I believe primarily with reference to leukemia. Our other speaker is Dr. June Yang, who I had the pleasure of recently having lunch with halfway across the world in Singapore. He is currently Vice Chair of the Department of Pharmacy and Pharmaceutical Sciences, and also has a particular interest in pediatric leukemias. So welcome, Drs. Yang and Cruz, and we very much look forward to what you have to tell us about precision medicine and functional precision medicine efforts at St. Jude's.
Great, thank you, Tony. I'm going to go ahead and share my slides. Let's see. Can you see my slides?
Yes.
Yes, they're not in presenter mode. There we go. That's presenter mode.
Okay, perfect. Great. Well, thank you again, Tony, for inviting both of us to give a presentation today about our effort in functional precision medicine here at St. Jude's. I think our talk is probably going to be a little bit different from the ones that you have heard last year. Ours will be a little bit more practical in terms of our thought process and all the practical steps that we are hoping to implement functional precision medicine here at St. Jude, especially in terms of drug sensitivity profiling and coupled with our genomic profiling efforts. All right, so this is the outline of the presentation. I will start by giving a very, very brief introduction about the current paradigm of precision medicine and why, especially in cancer, why there's a huge need for pharmacotyping or drug sensitivity profiling. And then I'll talk about our efforts in doing functional precision medicine or drug sensitivity profiling here at St. Jude, focusing on LLL in the past 25-something years. Talk about the evolution of different platforms and things that we have learned along the way. And then I'm going to turn it over to Chris, and she's going to talk about our efforts in the developing CLIA-certified pharmacotyping assay and our plan for clinical implementation as well. So many of you know already the current paradigm of cancer precision medicine has a very linear mentality. We almost always start with the sequencing, with the idea that if you sequence a patient, he has mutation A, he will receive drug A, mutation B, drug B, so on and so forth. So it's a very linear relationship between the genomic abnormality and the drug they're supposed to receive. So this relationship, although seems beautiful, only works in a very small fraction of patients diagnosed with cancer. Some of the examples are very well known. BCR-ABL with the imatinib, dasatinib, and various generation of ABL inhibitors that worked really, really well for pH-positive LL, CML, etc. And then there's the EGFR inhibitor, ALK inhibitor, imatinib, dasatinib in GIST as a c-KIT inhibitor, and so on and so forth. As I said, when it works really, really well. But for vast majority of the patients, this linear relationship doesn't necessarily hold true. So I actually stole this from a paper that's wrote by Tony quite a few years ago, looking at the NCI match data. I think this data is probably several years old already. At that time, about 6,000 participants, about 38% of them matched to a target therapy, and 18 actually had access to therapy assigned to a trial arm. For the patients who end up receiving target therapy based on their genomic profiling, the overall response rate actually ranged from 2% to 38%. As a matter of fact, the vast majority of the patients are on the lower end of the spectrum with a fairly low response rates. And of course, there are many, many patients with the genomic abnormality, with no clear cancer mutations. Their cancer genome is very, very silent. And then you have a lot of patients with a clear driver in terms of the pathogenesis of their tumor, but then the oncogene, for example, TP53, is undruggable so far. So even though you have a genomic finding, you don't necessarily have a therapy to match with it. And what I hope to tell you, and also motivation for our work here at St. Jude, is the observation that many patients may respond to a specific therapy even without the purported genomic driver. So for example, dasatinib is a multi-target TKI, can hit multiple different signaling molecules that are important above and beyond BCR-ABL. So patients don't have BCR-ABL may also respond to dasatinib because of the activation of other things that are hit by dasatinib. So what I'm going to do now is to talk about some of the things that we have been doing here at St. Jude on the research front. And then, as I said at the beginning, I will turn over to Chris to talk about the clinical implementation of this. So the disease I'm interested in is acute lymphoblastic leukemia. For those of you who are not familiar, this is the most common cancer in children, about 3,500 new cases every year in the U.S. By no means, this is a big cancer. It's a very small, right, compared to many other cancer or tumors that many of you work with. This is a malignancy that characterized by this uncontrolled proliferation of the undifferentiated lymphoblastoid cells that you can see under the microscope, compared to the normal bone marrow where you have a plethora of heterogeneous immune cells in hematopoietic cells in the bone marrow. So we always talk about ALL or acute lymphoblastic leukemia as a very successful story in our battle against cancer. In 1962, when St. Jude first opened here in Memphis, the cure rate of ALL in children is about 10%. So we lose about 9 out of 10 kids to ALL. Our most recent trial that was finished in 2017 was total 16. And there we achieved an overall survival of about 95%. So we're now curing 9 out of 10 kids with acute lymphoblastic leukemia. So you can say this is a true success because we completely flipped the statistics from losing 9 out of 10 to actually curing 9 out of 10 kids. But as much as we want to claim the credit, I think it's fair to say that the improvement in survival had very little to do with laboratory investigation or laboratory research. It's primarily driven by successive clinical trials, pragmatic trials where pediatric oncologists adding more and more drugs and testing different combinations of drugs that have been approved for a long, long time. I don't have the annotation here, but for example, from total 1 to total 5, where we actually use a limited number of cytotoxic drugs. In total 5 and total 6, we started mixing them together, 6-MP adding together with Kristine, with glucocorticoids, and adding them together actually made a huge difference. And then in the late 70s, and we started using things like asparaginase. So every time we introduce a new drug, it also lead to a significant jump in cure rates. Again, these are pragmatic, empirical clinical trials, not necessarily driven by a scientific discovery of the biology of acute lymphoblastic leukemia at that time. So here are the drugs that's been approved to treat ALL over the years. Many of them are very, very old. Mercaptopurine and methotrexate approved in 1950s, and then prednisolone immediately after that. As you can see, most of the drugs that we use today are at least 30, 40 years old. And by using the same old drug that was approved a long time ago, we're actually able to achieve this significant improvement in cure rate, which is pretty remarkable. And also, I think, as I said, a lot of credit goes to the pediatric oncologists who ran those empirical clinical trials, just testing different combinations of drugs approved for a long time. So this is the way that pediatric ALL patients are treated. It's rather complicated schema. We usually start with the induction therapy, including somewhere between 4 to 6 different drugs. And then we try to consolidate the remission, adding additional drugs, including glucocorticoids, high-dose methotrexate, asparaginase, etc. And then we have intensification, where we reintroduce some of the high-intensity chemotherapy used for induction, just give them more time. And then we use maintenance therapy, where patients were kept down for about 1.5 years with just simple chemotherapy regimen, methotrexate and mercaptopurine for the most part, and then adding pulses of Kristine glucocorticoids. So when I first came to St. Jude 2006 as a fellow, I was taught that there are about 5 or 6 different subtypes of acute lymphoblastic leukemia in children. By immunophenotype, there's about 17% have a T cell immunophenotype, 83 of them are B cell ALL, and then within B cell, they're hyperdeployed, about 25%, with more than 51 chromosomes in the leukemia genome. And then you have the big translocations such as ETV6 RUNX1, about 20%, and then 119, 11q23, 922, which is the Philadelphia chromosome, so on and so forth. So it's it's heterogeneous, but manageable with a few different molecular subtypes and also immunophenotypes as well. But so this is the new sort of a classification for B cell acute lymphoblastic leukemia around 2020. Now there are 23 different molecular subtypes of BLL. So we went from about 4 to 23 in the span of 16-something years. And this is a T-SNE plot. Each dot is a patient. Each color denotes a different molecular subtypes. As you can see, these 23 different buckets are classified on the basis of a number of genomic features, fusion genes, chromosomal translocations, gene expression profile, mutations, copy number changes, and so on and so forth. So it goes without saying that this is incredibly complicated for even people like me who study ALL every day, not to mention the treating physicians who actually see patients. A big question we almost always get asked is, now you have this highly complex genomic landscape of ALL. How does that impact the treatment of this disease? Do you still use the same old drug that you used before for 50, 60 years, or you can actually use this genomic information to individualize therapy and implement precision medicine? So the quick answer to that is no, not really. we know a lot more about leukemia biology, but not everything that we learned in the past 10, 15 years can be translated into a new therapy or even ways to improve the therapy that we have already. So, so in order to understand, I think because I'm in pharmaceutical sciences department, and obviously what we're interested is how genomics can impact drug response. And this is not necessarily a new topic or a new concept pioneered by myself. Actually, Bill Evans, who was the former CEO at St. Jude and also in our department, he started doing this back in the late 90s, the early 2000s. So he was in collaboration with Rob Peters in the Netherlands. They started doing leukemia drug sensitivity profiling, primarily focusing on four cytotoxic drugs, glucocorticoids, vincristine, daunorubicin, and asparaginase. These are the four drugs that we use during induction, highly effective. And they were doing drug sensitivity testing against those four drugs individually. And then Bill was doing microarray gene expression profiling in parallel. And then they tried to marry these two together and try to come up with a gene expression profile that will predict sensitivity to any one of these four drugs. And then they look at, they ask the question, if somebody is predicted as sensitive or resistant to a drug by gene expression, does that patient also have a worse or better outcome in terms of EFS OS? And as you can see, this is a paper that they published in New England Journal in 2004. There's just a huge difference by disease-free survival based on the gene expression driven prediction of drug sensitivity. So that actually worked really, really well. Since that paper, Bill has continued to focus on this topic, looking at specific genes in terms of their role in the response or resistance to specific cytotoxic drugs, and he has published many seminal work after that. So the method that has been used, especially under Bill's leadership, was MTT based assay. This is around started it's 1990. We learned that from Rob Peters in the Netherlands, and we kept doing that until almost about 2015. And the method is pretty simple. You take the leukemia cells from patients. You see them in a 96-well formats, and you actually put quite a bit of leukemia cells in there per well. And then you add your drug, incubate it for 96 hours, and then you add MTT, and then you look at the absorbance and use that as a measurement of survival or viability, and then you construct a dose response curve from that. So this is a method that's been used by many others. I think a lot of people still use this. They don't necessarily use an MTT, they use CTG and some other variation of that, but the concept is pretty much the same. So around 2015, we shifted away from the MTT-based assay to a more flow cytometry-based platform for two reasons. Number one is that the MTT assay, especially if you just take the leukemia cells out from a frozen vial, the viability is actually really, really poor. You lose like 50% of the cell, for example, and which is really not good, especially for some of the collaborations that we have. We only have access to frozen cells. So we decided to start adding the mesenchymal stem cell, or MSC. This is a feeder layer. We put it in the bottom of the well. And then on top of that, you add your leukemia cell at the same density as we did before for MTT assay. Again, incubate for 4 hours, and then we use MaxQuant, which is a plate format flow machine to look at first of all GFP, which we'll use to label stroma cell, and then we look at the CD marker, CD19 for B cell, CD3 or CD7 for T cell leukemia. And then we focus on the leukemia cell, look at the Nixin-5 and DAPI staining to indicate the degree of apoptosis. So this is the assay we used for about 5 years, working really well. And I want to show you an example why this is particularly useful. This particular leukemia sample, TLL came in, all of them are CD7 positive, but then on flow cytometry, you can tell there are actually two different clusters. within the CD7 positive blasts. So there's this is a forward scatter and side scatter. And then you can look at the apoptosis of each population in the presence of different drugs. So the first plot here is the dasatinib. Y-axis the viability, x-axis the concentration. The bulk population is purple, the line in the middle, and then the clone 1 is the one on the top, which is the blue curve showing relative resistance to dasatinib. And then you have the second clone, which is the lower one right here, indicated by a red curve, showing relative sensitivity to dasatinib. Of course, if you look at the bulk, is the average of the two. And this is also true from ponatinib, sensitivity of the second clone, but resistance of the first clone. But then if you look at venetoclax, there's no difference between the two. Both clones are equally sensitive. And then this is a FLT3 inhibitor, again, showing some difference. So this is an example of how this flow-based assay is actually quite informative in terms of a clonal heterogeneity, particularly for drug response. And clearly there's a variation from drug to drug. So around 2000, we decided to move to be creative again, or try to test a new different platform, moving from flow cytometry based assay to imaging based assay. One of the biggest motivation for this is because we're using a lot of cells for the flow cytometry assay, about 18 million cells for 8 drugs. And it also takes a lot of time, the sample prep process, the processing, and also the flow, even with Max Quang, took about 2 hours for a 96-well plate, which is quite long. So switching over to an imaging system in collaboration with Jean-Pierre Volkran's group in Switzerland, this is a 384-well format. We can test up to 44 drugs using only 16 million cells. So the throughput increased quite a bit. The sample processing time and also imaging time also went down quite a bit compared to what it was before using flow cytometry. So this is our workflow. I think Chris will talk a little bit more about this. We used a lot fewer cells per well, and we still use a stromal cell co-culture system, still 4 days, and then we use a fluorescent dye to measure viability. And then this is the imager that we use, which is operated CLS from a PerkinElmer. This is what a sample image actually looks like. We later on actually figured out we don't have to use 2 different fluorescent dyes. We can actually just use 1 fluorescent dye because the stromal cell and leukemia cells look very, very different under microscope. You can actually determine which is leukemia, which is the stromal cell, just by the size and also the intensity of the fluorescence from the nuclei to tell. So this is the workflow going from identifying the outline of the nuclei, looking at which is leukemia, which is stromal cell, and this is sort of the virtual flow, if you will, looking at 2 different populations. And then you can focus on your leukemia population and look at the viability based on the fluorescence stat, and then construct the LC50 curve. So accumulating, all the data, 3 different platforms, MTT, flow cytometry, and also imaging, we put together this huge cohort of ALL cases, about 1,000 patients, 50% pediatric, 50% adults, doing pharmacotyping and also doing comprehensive genomics. The idea is to construct the gene drug connectivity map so that we can use this information to guide the development of the next generation of ALL trials here at St. Jude, and also elsewhere, especially in collaboration with our adult colleagues. And this effort is led by many of us, including myself, Bill Evans, Mary Relling, Charles Mulligan, and others are not showing here. So right now we have about over 1,300 cases. Newly diagnosed is the majority of them. We also have almost 160 relapse refractory cases. This is the list of drugs we're testing. Cytotoxic drugs are the ones that we currently use to treat pediatric ALL. And then we also include a lot of targeted therapeutics, especially TKIs, many of them representing different or hitting different signaling transduction pathways, BH3 mimetics. We recently included navitoclax as well, HDAC inhibitors, proteasome inhibitors. We added carfilzomab recently. And also we have antibody drug conjugates, inotuzumab, which is a clindamycin conjugate with CD22 antibody. So this is a huge data set with 67,000 data points. So this is another way of showing the data. As you can see, each vertical column here is an individual. We have patient characteristics showing as a circles, age, sex, ancestry, molecular subtype based on genomics, and each dot here is the drug. So you see the cytotoxic drugs are the inner circles. We have them almost completely filled because we've been doing cytotoxic drugs for a long, long time. And the molecular targeted drugs are on the outer rim. The dots are more sparse because we started doing them more recently. As I mentioned already, we're doing a lot of genomics, different type of genomic, molecular profiling for this cohort of patients as well. So the way that I think about this is we have the drug sensitivity data sitting in the middle, and then we have different layers of genomics, whether it's whole genome sequencing, RNA-seq, ATAC-seq, so on and so forth. And then with this core data sets, we can do a number of different things. For example, we use the genomics to define molecular subtypes of ALL, and then we look at the correlation with the drug sensitivity on individual level. And then we also look at the specific genomic event. It could be a mutation in RAS gene or TP53 or Notch pathway, and we can look at specific gene fusion, BCR-ABL being one of them, and there are many other chromosomal translocation events in leukemia that lead to fusion genes. We can look at them individually, see how they influence the sensitivity or resistance to a specific drug. We can look at structural alteration. These are the copy number gains and losses. And we also are doing methylation profiling to look at its association with drug response. From RNA-seq, we can infer alternative splicing, and we can look at each different isoforms of transcript to see which one is associated with the sensitivity or resistance to a specific drug. We're using RNA-seq. We're also doing analysis right now to infer leukemia maturation arrest and developmental state to see how that can modulate sensitivity or resistance to specific drugs. And of course, the list can go on, and many of these are ongoing work, and we can add more and more molecular profiling techniques and features that we can use to correlate with drug sensitivity. So the way, like I said, I think the goal here is to functionalize ALL genomics or omics, if you will. I mean, whatever term people use, because a lot of people are doing omic profiling of various different cancers. I think what we're trying to do is to add the drug response as an extra layer, and hopefully that will help us functionalize the genomic and molecular features that we learn from different profiling techniques. And hopefully that will eventually translate into new therapy or more personalized approach to treat patients. So now I'm going to turn it over to Chris for her to tell you a little bit more about our work in developing the CLIA certified pharmacotyping assay, how we envision that will be rolled out in clinical practice. Chris, should I stop sharing and turn it over to you?
Yeah. If you'll stop sharing, I'll go ahead and share my screen.
Okay. Actually, before we do that, if anybody have questions at this point, I'll be happy to answer them. Or if you don't, we can wait until the end.
Just a quick question, technical question, June. When you do your sort of image-based assay of your ALL cells, is adhesion a concern? And how do you get good two-dimensional imaging of what is essentially a non-adhesive cell type?
That's a very good point, Tony. So in our experience, if we have the stromal cell at the bottom, all the leukemias, they will settle. They will sit on top of the stromal cell. And when you image, they're almost right on top of it. So they're not floating around. It's very easy to image.
Can you? Does any of your protocol include a rinsing step? Do the ALL cells persist through a rinsing step?
we don't rinse. If you rinse, you will be much more difficult to do. I think if you want to rinse, you probably will be better off using a flow-based assay. All right, if there are no other questions, I'm going to turn it over to Chris now.
Thanks, June. I'm pulling up my slides now. Okay, great. Dr. Latai did the introductions at the top of the hour. I'm Kristine Cruz. I'm the Director of Research Operations at St. Jude. I work closely with June to generate the pharmacotyping data for our assay. So I'm going to talk about some of the aspects that went into developing this assay for pharmacotyping, which again is measuring drug sensitivity of a panel of drugs in primary patient leukemia cells. So in this image on my first slide, I'm showing the fluorescence image that is generated with our assay, with the red arrows pointing to leukemia blasts from a primary patient sample, and the white arrows pointing to the stromal cells that are co-cultured with them, which are much larger. So talking through what we need to complete the pharmacotyping assay for our patients, I mean, we start with leukemia cells, which we can get from either bone marrow or peripheral blood. We really need to just plate 16 million cells, and we test 40 drugs on 2 different 384 well plates. And so one thing to note about the co-culture system, we do need to know 24 hours in advance of when a sample will be collected and received in our lab, because we actually plate the MSC co-cultures the night before and let those seed overnight. We want the MSCs to be 100% confluent the next day at the time that we add the leukemia cells and then the drug concentrations as well. June kind of talked through the fluorescence imager assay. It's important to set up a way to discern these 2 cell populations in your field of view. And we created an analysis pipeline which trained on the nuclei size and intensity to be able to do that. What we're ultimately looking to do is to test increasing concentrations of each drug and generate a viability curve so we can calculate an LC50 for each drug for each individual patient. And so thinking about some of the more characteristics of what we need to work with, we can work with either fresh or cryopreserved, and we validated for both types of samples. We prefer fresh samples, and we do have some external collaborators, and we've had success with shipping these samples overnight to our lab. We've validated testing with fresh samples that are up to and including 4 or 5 days old. Obviously, you lose some live cells each day, but we have had success with even shipping some samples from external sites. We can also work with cryopreserved samples as well. We prefer fresh because we have a higher percent QC pass rate working with fresh cells versus cryo. But this pharmacotyping data has been really crucial in combining with genomic data, as June outlined for you, and really feeding our pharmacogenomics research discovery pipeline. But we actually envision a way to use these results actually to help further stratify treatments for patients in the context of clinical trials, and even to kind of design a treatment regimen for an individual patient. And so towards that, we spent a lot of effort over the better part of this past year taking our research assay, which we have a lot of experience with, and migrating it to the clinical setting. And so I'm going to talk about that, sort of the good, the bad, the challenges that we've had with doing that. So my screen is a little funny. So we have the ability to expand to using these results for patient care. So actually making this a clinical functional precision medicine assay. So we have done the extensive validation that's required for CLIA to be CLIA approved. And I'm going to talk through kind of what those considerations are. And I realize that many of the participants here on this call, I'm looking at each of you guys. So there's a lot of US-based people, and there's also some non-US-based people. So just to take a minute to kind of get everyone to understand sort of where we're starting from. In the US, we talk about CLIA approved tests, but does CLIA really approve the tests? And not actually. So what happens is labs like ours can develop our own tests, and a laboratory developed test is an in vitro test that's designed and used within a single laboratory, like our pharmacotyping test is. So currently, in the very current state, the FDA currently practices what is called enforcement discretion with laboratory developed tests. So that is a laboratory developed test like ours for use in our patients does not need to be FDA approved currently. But labs do have to comply with CLIA regulations and validate these laboratory developed tests. And then when you report them in the patient report, you just need to include a disclaimer that this is not an FDA approved test. And so that is the route that we're taking as we have developed our clinical assay. And then just a note about CLIA. So CLIA is the Clinical Laboratory Improvement Amendment. And all labs that do report tests for clinical patient care do have to have a CLIA certificate. But actually the body that helps with doing that is the College of American Pathologists, or CAP. So you hear about CAP coming to do an inspection, and they're really the deemed organization that's the feet on the ground to make sure that CLIA regulations are followed. So that's why you hear about CLIA and CAP and inspections and surveys. But just to point out that many of you are familiar or have been reading that all of this could change with a pending FDA proposed rule that was proposed in September of 2023. And the FDA is proposing to classify laboratory developed tests like our test as medical devices, and as such, they will require FDA approval. And that proposed rule is in draft phase right now, unclear on the future of it or how soon this is really going to happen, but it's going to affect us and many other labs that are doing laboratory developed tests. So just to walk through the thought process to take a research test like we had with a lot of experience with it and roll it into the CLIA environment, there are certain requirements for a clinical test, which both the lab has to meet and your documentation and your records have to meet these requirements, as well as each individual test. And so I've highlighted here that there's a need to establish the method performance specifications, to set calibrators and verification procedures, as well as control procedures and comparing test results. And so we've had to work through each of these requirements for this established test, which was already very successfully run as a research test. There's many more things to consider for the method validation to meet the CLIA requirements. And so I've listed them here on this slide. So you need to think about our pharmacotyping assay is considered a high complexity procedure. You need to think about establishing precision or repeatability. And so to address this, we ran multiple intraday, which is several tests in a single day, and interday, so over days and weeks, to establish the repeatability and to look at the variance of each run. And then also there's a requirement to establish accuracy. And the typical way to do this for a chemistry test is to compare to a standard reference material. So we're working with a biological assay where each sample is really unique to itself. There is no standard reference for this assay. And so to address this, we, in our validation procedure, we compared to the previous MTT dye-based results, which we had hundreds of, and flow cytometry-based assay results as our reference to address that. We also had to establish all the things that you would establish for a quantitative assay, the reportable range, the reference interval. We had to establish limit of quantification of how many cells that you can pick up, any interfering substances, as well as other, we did extensive freezer stability of all of our drug stocks, and just document over a series of many months, really. And so something that's interesting, this assay is very unique to a laboratory developed test because it's a biological assay. We're working with living cells. We have to decide which CAP checklist to follow. So CAP publishes, I think there's probably 15 or 20 different categories of tests. And this biological assay didn't really fit entirely into any of them. It's a little bit hematology, but it's not really a hematology test. It's definitely not a microbiology, even though it has some aspects of microbiology work. So we ultimately decided to that it fits best in the chemistry and toxicology checklist. But that also creates some challenges because I copied here a snippet of the CAP checklist for a chemistry test. And you really have to decide if your test is a quantitative test or a qualitative test. So a qualitative test, in thinking about our pharmacotyping assay, we're measuring sensitivity of a patient's cells to a panel of drugs. So it could be qualitative if you can easily say that this sample is sensitive or resistant and just categorically set it. But you lose a lot of information by not reporting that LC50 value. So we quickly moved away from that and realized we need to report the LC50 values, and this is a quantitative test. And so by the letter of the CAP requirements, you need to have 2 controls at 2 different concentrations for a chemistry test, which was a little bit of a challenge to set up. But the way that we addressed this was we validated 2 commercial ALL cell lines that have differing sensitivity profiles, and those are our controls. And so just to give you a snapshot of how that works, we selected 2 ALL cell lines that are commercially available. The first one is BAL1, and this is a B-ALL cell line. And so just to show you, we repeated replicates of all of the drugs that we're going to test and report. And I'm showing here some results for ponatinib. This is 5 different replicates where we plated in duplicate ponatinib with this ALL cell line on several days. So this is showing 5 different days of experiments. And then to contrast that, our second cell line is called ALL-SIL. This is a T-ALL cell line. This was created from a relapse sample from a 17-year-old boy whose cells had an ABL fusion gene. And so this one is expected to be very sensitive to ponatinib. And in our hands, we were able to show that it is very sensitive to ponatinib. So we can contrast those 2 and essentially have 2 different control points for your control line. And so then what we did with that, to illustrate for you, on the left-hand side is the BAL1 control charts of 22 replicates measured over 22 different days. On the right hand is the same for ALL-SIL control charts. And so we have charted both on the left panel is the AUC. So this is actually the area under the effect curve estimates for each day's experiments over 22 days, with a moving average shown as a red line that's moving along. And then next to it is the LC50 values themselves measured over 22 different days. So we're able to capture those in the course of our validation and show the variability and really know what the performance is for those 2 cell lines. And something that is quite clear when you're looking at the ponatinib results. So if you're reporting an LC50 as your measure, and you have a very sensitive sample like the cell line for ponatinib, you're not going to have an LC50 estimate because it's actually not crossing the 50% viability point. So you're showing this control chart, which shows very sensitive across all the way. And that's where actually the shape of the curve can be captured and quantitated a little bit better by using the AUC control charts. So we're looking at both of those. And so just to show you that for one of the cell line controls, which is BAL1, we did 20-day replicate plots for all of the 20 drugs that we're going to report, and we can show the variability across those days and really capture those measurements and variability estimates. So then a second consideration, just talking through the practicalities, it's important for us to discern between the MSC cells and the leukemia cells in a sample. And so we are able to use machine learning to automate the count of these cells, which is important for the viability estimates. And this has greatly improved the robustness of our methodology. So we worked over a series of months and trained each improving machine learning model to actually recognize and train and call a leukemia cell, and then do our count that goes into the viability estimates. And we were able to validate the manual, which is a human-based process of selecting which ones are leukemia cells, versus the machine learning model. So the manual results for viability are plotted here in green, and the machine learning with the final model that we went forward with is shown here in red. And we saw a very good correlation, which we are able to show using looking at comparison plots. And this is a representative example of selinexor results for each of the viability measurements by the manual method versus machine learning method. And we showed very high regression values across all of our drugs, with the LC50s being calculated in that manner. And we also were able to look at a Bland-Altman plot, which clinical laboratorians really love their Bland-Altman plots. This is a way to look at systemic bias across comparing two different methods. And we are able to show very low bias and very tight correlations for each drug. And so we were able to move forward with this machine learning process, which really enhanced our pipeline. So another thing to take into account for a CLIA, for the CLIA requirements, is the need to report clinical validity of your test. And so we were able to address this by actually reporting on patient results that we've run, and selecting patient results that exhibited a certain subtype. And so in this example that I'm showing, we tested 30 patient samples. 3 of those samples were positive for the TCF3 PBX1 fusion gene, and they're expected to show more sensitivity to dasatinib. And so we're able to show a lower median LC50 for this patients with this subtype versus 27 patients that don't have this fusion. And we're able to do this for several different markers and to show that the sensitivity falls where we would expect it to fall as a measure of clinical validity. And so getting to how we actually will run these samples. So so when we receive a patient sample to be run, we actually will run 4 plates on each testing day. So we have 2 plates of patient samples. There are 384 well plates. So as a research assay, we were assaying 2 plates, 40 drugs total. And so what we've done is taken forward plate 1 as our clinical plate, and this is what we can report clinically. We're also still running the 20 drugs on our research plate. We'll continue to run them, continue to collect those research samples in our research database, but not report them for patient care. And then each time we run a sample, we have to run 2 plates of each of the each of the control cell lines. And so it's a total of 4 plates that we run with the drugs that we plate shown here for your viewing. And then just to think about how we're going to report these in a patient's medical record. So we're working with our information sciences team to build actual clinical report in the electronic health record with a test named tumor pharmacotyping. And so the way that we're displaying it is every time that there is a test with a panel of 20 drugs clinically, each of those drugs will be reported in the report with the patient's LC50 value reported there. And what we've chosen to report as our reference interval is based on all patients that have been run before this patient. So we're calling our 25th to 75th percentile our reference range. We also have the ability to show what the median has been observed. And so that just to illustrate for you for this patient's results, the LC50 for bortezomib, that the viewer viewing these results can quickly see where this bortezomib LC50 lies, it's higher than the median, it's higher than the 75th percentile, and it's relatively resistant compared to all the patients that have been run with this drug in the past. So this is how we're envisioning reporting this. And then just to follow up with really just kind of, we thought it was still important to mention, again, this proposed FDA ruling for LDTs. So when we started our CLIA validation, we decided to move this forward as a lab-developed test and not as an IDE, because it's not going to be used alone for any patient decisions. It's going to be used in combination with genomic results. So we built it, we began building it as an LDT. This draft ruling came out in September of 2023, which might change things really in the somewhat near future. So this proposed ruling means that this test and all laboratory developed tests will need to be FDA approved, which really could affect our progress going forward. And that's something that many of us that are looking to take our research test into the clinical realm are really going to have to think about. So we just wanted to end this. June and I want to make sure that we thank the team, the huge, wonderful team that does our pharmacotyping and works with our pharmacogenomics research, who are mentioned here. And we're really happy to continue a conversation or take any questions that you have.
Great. Thank you, Kristine, for a really detailed and very, very important discussion. I'm afraid I am going to monopolize you for just a bit and then open up the questions, because I think there's some very important points you made up. First, just a quick technical question. This issue of sample handling is something I think we often gloss over a little bit. Doesn't appear you are. So I want to ask a specific question. Have you ever done a series of comparisons of fresh samples that you have then subjected to freezing and compared the results of your pharmacotyping?
We have. Yes, we have. Very intensely. We have been able to test a sample fresh and then cryopreserve it, thaw it, and retest, and shown a good correlation between those two.
Great. So I want to start by emphasizing what you said about the potential problems with the FDA getting involved. And I want to summarize for the people here. This is something that we at the SFPM may, as an organization, want to get part of. So I encourage you to think about it and think about how we might help. I have found it to be very difficult. So we have an assay we're trying to get an IDE for a clinical trial. It's one in which it will be part of the, it won't be the sole like algorithmic decision-making tool. We will of course include other elements to make it. And we chose to go the IDE route, which may or may have not been wise, but we were directed. We also needed, there were experimental drugs. We were directed by the IND side of the FDA to go for an IDE. So that's what we ended up doing. A real challenge is it isn't a can-do relationship with the FDA when you're doing something novel. They are not incentivized to make this happen. It is new, and for them, I think, a risky work, and they're all worried about riskiness. In the case of our case, we're dealing with relapse refractory AML patients that have a median survival well below a year. And it is, of course, absurd to be thinking about risk in that circumstance with regard to assigning therapies. The biggest risk is they won't get a therapy and they're going to die in a week. What I'm very concerned about in addition is they simply aren't manned for this. The people there that we encounter, I think, are all trained to, evaluate assays for serum levels of sodium or something like that. But these complex assays, they bring that same thinking to, and it's not a problem-solving interaction. The other thing is the communication is terrible. You submit something to them, there's radio silence for 60 days until your next meeting, and then 20 hours before the meeting, there's a doc drop, and you have to go over this, and that's your one chance. Then you have like an hour to interact with them, or half an hour or something, and then boom, that's it, you're done. And they move on to the next. And I think a lot of it is because isn't their fault, it's because of staffing. But if they're going to take this huge bite out of
No.
LDTs. I would like to know, are there resources provided for essentially tenfold increasing the staffing at the CDRH, which takes place of this? And this is potentially a huge problem.
Many of us have that same concern, right? Where this is going to, you know, maybe 100-fold or 1,000-fold increase the number of approvals that they need to be staffed to do, and that is a huge issue with this ruling.
Right. But it's, I mean, the less the FDA is involved, the better. But I understand there has to be some regulation. So I have a more specific question. Can you get into, so ultimately, if you're doing this in a everyday clinical setting, you don't need to involve the FDA, at least up until now. That may change with the new LDT rules. If you are running a clinical trial, and especially using novel agents, you sort of have to get the FDA involved at the IDE level. So can you clarify for me something I don't quite understand? Is it your local IRB that's going to tell you whether you need both a CLIA and an IDE, or one or the other?
Yes. At the time that we would implement this in a clinical trial that is returning results, you know, for an example, to stratify patients to the treatment arm. We'll work with our IRB to how we interpret the need for an IDE. But what we're moving forward with is that if it's not the sole measure that you're using to make that stratification, if it's not the only measure, then it won't need to have an IDE. That's what we're moving forward with the interpretation.
Okay, so what sounds fine to me, favorable, but what do you base that distinction on? Is there some precedent for that distinction allowing you to escape an IDE?
So Tony, my understanding is if you use this as a piece of information, it's almost like a molecular tumor board.
Right.
So you use all sorts of information on your molecular tumor board, and none of them, or not all of them, are FDA approved IDEs. So this is the way to approach it. And this is actually what, based on our interaction with other people who have been doing this, that seems to be the convention.
Very interesting. So is that accepted? So is that determined? I'm just trying to figure out who gets to make that call that's good enough. Fine with me. Mind you, I'm not trying to get in the way. I would like to do this. Is it your IRB that is the ultimate arbiter of that, or who?
I don't have a clear answer for you right now. It's local IRB. But then who knows? The FDA might come and say, well, no, you can't do this. Your IRB doesn't get to decide this. But IRB is our point of contact right now, our IRB.
Okay, I'll shut up. I see a question in the chat, and then put up your hand if you have an additional question.
Tony, can I go ahead? Sorry.
Oh, go ahead, Diane.
Yeah. So my name is Diana Azam, and I have a lab at Florida International University. Thank you, Kristine, for such a thorough presentation on, you know, the challenges and the process that you have to go through CLIA. I can relate because I'm also trying to get I'm going through the validation to get this CLIA, and my test is a little different in terms of just, I mean, I don't do imaging, we do luminescence, but so we're going through the whole process, and I understand, you know, the challenges, but I did come across one very big challenge, and I don't know if you guys thought about that or even talked about that. So if you validate your single agents and you want to go ahead and test combinations, will CLIA allow you to test or tailor combinations based on the validated single agents? And what I understood is that you can't do that. So if you validate 40 single agents, you have to validate your combinations as well. And that's a major challenge, because you would think that at some point we want to be able to test different combinations, right, of drugs that have been validated by CLIA. So I wanted to hear your perspective, if you've even talked to anybody regarding that approach, because there is no way we're going to be able to validate, you know, 30,000 permutations of combination agents. So, and I've spent hours with people talking about this, and I've been hitting a roadblock. Yeah.
Well,
so
we have not had to think about that yet. We're only testing in singlets right now. So I don't know. I'd like to hear what, if you've talked with other people and are other people interpreting it as each combination is its own assay, which will need its own
Yes.
validation.
Well, can we learn from the microbiology community? Because in the microbiology community, my understanding, like, so of course, these sort of functional tests have been used and continue to be used as a gold standard for the last 60 years or something, okay? Now, my understanding is in at least the report that's provided to the physicians are 100% single agent, okay? However, the ultimate use in a lot of cases is in a combination therapy, say vancomycin plus some third generation cephalosporin, or maybe it's going to be levaquin or something like that. So there is precedent, it occurs to me, that we can refer to in assays being used to choose therapeutics where the assays are single agent, but the ultimate use is in combination.
That's true, Tony, but I mean, if you think about it, you know, as we know, we've seen that you put drugs together, they in different combinations, they act differently. Sometimes drugs are ineffective, a single, put them with another drug, they're effective. So if we really want to end up testing combinations on tumor cells, I think that's the best approach for functional precision medicine at some point to tailor treatments, right?
But I think you also made the point that opens you up to having to test 30,000 combinations.
Right.
And if you want to test combos directly, great. But I think our position should be to allow the assay provider to make that choice and not to have it mandated by a regulatory body. We should definitely avoid any hint that a regulatory body could mandate direct combination treatment. I also say I've seen people who actually tested this data. This was at a meeting a month or two ago, where they looked into that question, and roughly 80% of the value of combinations can be determined based on single agent information. And you may miss some information by not directly testing the double agents, but you get most of the information. It's rare. Basically, it's rare when you find a combination that has activity where there wasn't single agent activity.
Yes, but based on I'm just going to tell you based on my experience in my clinical trials, what we found is that we, you know, sometimes 4 drugs together have the same effectiveness as 3 drugs together. And when you're talking about these cytotoxic combinations, many of the physicians found it helpful to reduce the number of combinations of drugs. And so testing combinations and seeing whether 3 versus 2 versus 4 drugs are safer or is important. And I really believe that this has been a very, very helpful approach for physicians to tailor treatments to their patients.
Right.
And
so Diane, I think we're talking about 2 different things. I think there could easily be contexts, and you're describing one, where the direct testing combinations are useful.
yeah.
However, I'd like to bring it back to the issue of we do not want this to be a regulatory mandate.
Well, so far, based on my conversations with the regulatory people, it is a mandate. If you have certain combinations that you want to test, you're going to have to include that in your validation. You can't just have different combinations tailored for every patient if you only validated your single agents. The only one, after talking to them for many hours, the only thing that came out of that discussion is if we use an AI approach and validate an AI approach that will give certain combinations, and if we validate it, obviously, then maybe we could potentially use AI to predict combinations as an LDT. That's the only thing that came out of my conversation, but that's why I wanted to ask Kristine and about their experience.
Well, yeah, we haven't had to add that, but that's really interesting.
But Diana, just so we can publicly agree on this, I hope you're describing what you see as a reality, that they want to mandate these. Do you agree that our position should be to fight against regulatory mandates of testing of combinations?
100%.
Good.
I agree with you, of course. It's absolutely, if we can do something about it, then as a group, of course, we should.
So, but this is what I was hinting at. I think Kristine will agree. I think part of the problem here is we're dealing with regulators whose primary goal is to keep their jobs and avoid risk. And the easiest way to do that is to say no at every turn to almost anything. And anything new is especially liable to having people say no to it. That's a problem. I will also say it's worth reflecting that in the European experience, that maybe that can be helpful, they are not mandating this sort of testing. For better, for the worse, I think our European colleagues will agree that they have experience in some of these FPM trials at sort of imaginative combinations of agents when there's only been available single agent testing. So there is in the civilized world a precedent for not requiring that may help us in the future.
And I want to add to that, we have ways to use controls to basically show if combinations are synergistic, combinations are antagonistic. I mean, we can do those, we can get those controls. We can do all those validations, but we're not going to be able to validate 38,000 permutations and of combinations.
And also keep in mind that history for pediatric leukemia, you're always going to use the chemo backbone no matter what you do. So nobody, I mean, I think it will be absurd for us to test combination of a new agent with glucocorticoids, methotrexate, 6-MP, because you're going to use those drugs, whether you do functional precision medicine or not. So I think if you're combining two new drugs, there might be an argument for that. But with the cytotoxic drugs, it shouldn't be the requirement.
FYI, this is Chris. Sengen routinely tests drug combinations. Just published a paper where a patient actually took the combination, identified by single agent combined with other single agents. So it is possible. I'll talk to you offline about, Diana, about how the CLIA aspects of that, but maybe you're just, yeah, it's a shame you're running into a roadblock, but it should be solvable because we've solved it.
But do you test the combinations together without validation? You only validate the single agents?
Yeah.
Yeah, okay, that is doable, but I'm saying if we really want to test all the combinations on the
Well, we don't do all of them. We just pick ones that are smart because you can't do 60,000. You just pick one drug that's interesting and test it against the other panel or two. That's how we do it. Does that make sense?
Yes.
Yeah, okay. Let's talk more later if you want to follow up on that. Please, we'll get you through that hurdle because it's solvable.
Okay, I think our enthusiasm for this topic has brought us well past the time of conclusion. I think we'll need to continue this. This may be something that we'll have to have a more concerted effort at and maybe this organization can help with this. I see problem ahead. I think, of course, you always want to rebel against, you often want to rebel against regulation. I'm very, very concerned that they will have the resources to actually perform this regulation that they're reaching for. And that does, the federal government is a behemoth and it's very hard to move, but I think this is a potentially very serious issue that we need to address. Finally, I want to say that some of the additional frustration that I find from this is that genomic precision medicine was subject to far, far less of this type of regulation. That, frustrating as it is, we should also remember that that is a potential precedent that we can use to our advantage. Sort of the idea I always have is we don't want special treatment, just regulate us like you regulate the genomics people. Okay, with that, thank you, everybody. Renew your memberships, and thank you very much, June and Chris, for some very excellent talks. Thanks. Bye-bye.
Thank you
all.